Hi everybody, and welcome to our webinar today. This webinar is the last in our series of webinars this year. In fact, it's our 9th webinar, so it's been a busy and a long but good year. Today, our main speaker will be Dr. Christoph Krafft. He's the head of Raman and infrared histopathology at IPHT in Jena in Germany. And I'm Dr. Mustafa Kansiz. I'm the Director of Product Management and Marketing at Photothermal Spectroscopy. So the main talk, so that's Dr. Krafft's talk, will be all about live cells, tissues, and single-cell bacterial analysis with Submicron IR, so that's O-PTIR, together with simultaneous Raman spectroscopy. Or comments or suggestions, there's a chat box on your screen there if you want to enter questions, and we'll do our best to answer that at the very end. So really, without any further ado, let me jump straight into it so we can get to Dr. Krafft's talk as soon as possible. So really, the outline, I'll take you through a little bit about vibrational spectroscopy and some of the limitations as it stands today with FTIR and Raman. I'll then introduce to you how O-PTIR works at a very high level. I'll give you a snapshot of some applications, predominantly based around publications. And I'll speak through those pretty quickly, so I hope you're sitting down, and then I'll pass the baton over to Dr. Krafft for his part of this webinar. So I assume most of you are familiar with vibrational spectroscopy. Suffice to say, it's all about vibrations of molecules, it's the name, and each of the peaks in a spectrum there correspond to a functional group, and it's been used for decades. It's been used across a whole wide range of applications, but today obviously we'll be focusing on life science, and that's perhaps the most exciting field and the area of vibration, I think, that's growing the fastest and has the most potential as well. But when it comes to infrared, so really you probably have all heard of FTIR and even some of these emerging Q-cell technologies, and they've been around for a while, but there are some fundamental limitations. The first and foremost, it's all about spatial resolution. If you're using a microscope, you generally care about small things, but because those techniques are dealing with long wavelengths, that's infrared wavelengths, your spatial resolution is typically limited to the sort of 10 to 20 micron area because the wavelengths are long relative to the visible. You can't do much about that. It's simple physics. Sample preparation can be difficult. Often the best quality data is obtained in transmission mode, but not all samples are amenable to be cut to sort of that 5 to 20 micron region. And to overcome some of these, there is ATR as a modality, as an accessory, but it requires intimate contact. Targeting can be difficult. You have to, if you have to make multiple contacts, there's cross-contamination issues. The crystal's expensive. It can be damaged, or worse, you can even damage the sample. And, here's an example of an ATR tip indenting and leaving marks across your sample. Furthermore, liquid nitrogen is often used as a detector to increase the sensitivity of the detectors, and some of them only have limited hold time, so you can't be doing really overnight measurements. But perhaps one of the biggest, and if not the biggest, I think, especially in the life science area, is these dispersive and scatter artifacts, sometimes called me-scattering. So in an ideal scenario, as I said before, the best quality spectra are obtained in transmission mode. So if you imagine that's a thin film of polymer, PMMA in this case, and if you measure that in transmission, you get this beautiful looking spectrum. Flat baseline, nice symmetric peaks, all looks good. But if you take the exact same material and make it into a bead and measure that in transmission, well, all of a sudden things don't look so pretty. You've got offset baseline, you've got a weird baseline, the peaks are shifted and split. And then as you change the size, the spectra change. Right? So it really goes to show how spectra in these more traditional FTIR or these emerging Q-cell based techniques can be very much particle shape and surface topology dependent, and not just chemistry dependent, because we assume the spectra to be representative of the chemistry, but in fact there is a component that is due to really morphology. On the Raman side of the equation, anyone who has used Raman, you will know that its arch nemesis is autofluorescence, and especially with biologicals, autofluorescence can be a major issue. That can be mitigated to some extent by going to longer wavelengths, but then you take a massive hit in sensitivity, and that's on top of an already limited sensitivity for Raman relative to IR. If you want to do imaging, you're forced really to do hyperspectral imaging. You can't do single frequency imaging, which is something I'll touch on in a little bit. There's a risk of photo damage or phototoxicity if you're working with live cells, because you're pumping in as much light as possible to overcome those sensitivity issues. And also Raman spectra can depend on the excitation wavelengths, not just the samples themselves, but even substrates have some dependency on the excitation wavelengths. So with optical photothermal infrared, O-PTIR, we've really overcome all of those, I think almost literally. What is O-PTIR? It's a pump probe optical spectroscopy technique where the pump is an IR laser and the probe is a short wavelength laser, typically a green or a 785. It delivers submicron infrared spatial resolution, so it's very much like Raman in that sense, but you're now getting infrared information with that. And it delivers FTIR transmission-like spectra. So that's spectra that look like they've been collected in transmission mode with an FTIR microscope, but it does so in reflection mode. So there are no distortions, no artifacts, no interference fringes. It requires minimal sample preparation, and the spatial resolution is infrared wavelength independent. Okay, just give me a sec. I'll start a quick video. How might this technique work? So it all starts with the microscope objective. It's a reflective Cassegrain style. Through that, we shine a pulsed infrared laser beam, and being infrared, it's fairly broad, maybe around 10 microns. At the same time, we're shining in our probe beam, the green. It's about 500 nanometers. The infrared generates thermal expansion as it's expanding, and that thermal expansion changes the way the green light is reflected or scattered back. Right, so as we're tuning our infrared laser from one wavelength to the other, we monitor the intensity of the green light reflected back. And it's from that we calculate out what is essentially a pure infrared spectrum collected in reflection mode. All right, when it comes to optical or O-PTIR measurement modes, we've pretty much got three. There's the single point and shoot mode, where if you imagine that's our field of view, you can go click, 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 and from each one of those points you'll collect a full spectrum. Each one of those points, mind you, is also about half a micron, that's about 500 nanometers. You can work in array mode, where you can do line arrays with minimum step size of 50, but I typically operate at around 100 or 250 nanometer step sizes. Or you can work in a more traditional mapping mode, hyperspectral mapping. I call that discovery mode, perhaps where you're not sure what's changing. And then you've got obviously a 2D array, and then with the spectrum it becomes a three-dimensional data cube. Or if you know what you're looking for, hence the term targeted imaging mode, you can work with single frequency. And this is just by virtue of the QCL, the properties of the QCL, which enable you to focus on only certain wavelengths and not every single wavelength. So that can speed things up dramatically. So in this slide, this is an important slide here. I'll try and compare and contrast traditional FTIR and QCL microscopy to Raman microscopy against a number of spectromicroscopy attributes, such as spatial resolution. If you cared about spatial resolution, typically, I'm guessing you'd go for Raman. That traditionally has much better spatial resolution than the traditional IRs. Hence, I've given that a red. If you cared about fluorescence, well, you wouldn't be going to Raman. You'd be going to traditional IR. Spectral sensitivity, IR would win that race, generally speaking. Hence, the speed of measurement also correlates with that. When it comes to libraries, commercial libraries, IR has typically about 10 times as many available or entries in these databases compared to Raman. It's also a lot more, I find, more spectrally interpretable. I've given that a green. If you want to work in reflection mode, which is that easy point and shoot kind of mode, Raman is great for that. Traditional IR is not. Reflection mode with IR just doesn't work well typically. If you cared about water vapor, Raman would be the way to go. If you wanted to work in solvent, maybe live cell experiments, again, Raman is great. Raman is typically compatible with glass, and Raman is also wavelength independent when it comes to spatial resolution. So when you look at that on your screen, it's no surprise that most labs will have usually an FTIR microscope, maybe, and also a Raman microscope, and you may go to one or the other depending on the needs of the experiment, or you may even have to use both instruments, in which case finding the exact same spot can be a bit of a challenge. But what we do, and this is my favorite animation, is we literally take all of those and put them into a single platform. That's the O-PTIR microscope. Now, I've talked a little bit about pump sources, so that's the typical QCL. I'll give you a little snapshot of what's happening there, some new developments there. If we take a traditional spectrum, the standard QCL that we offer is a 3-chip QCL with a 4th slot empty. Covers, as it stands there, 1800 to about 950. There is also a high wave number OPO option. That's an additional laser that does 36 to 2700. But what excites me is these custom QCL options. And my favorite, if one were to have a favorite QCL chip, that would be a CH region. That covers about 3,000 to 2,700, and you can combine that as the fourth chip in a single box. So therefore, your single laser unit can now cover the fingerprint and key CH stretching region. If you're interested, like some, in the silent region, where there are CDs, nitriles, alkyne sort of moieties there, you could do this sort of combination where you have the fingerprint and the silent region. But if you're interested in pushing out the long wavelength as far as possible, you can get down to about 800 easily, and then your fourth chip would be the one that would give you down to 800. On the probe or Raman excitation, of course, they're the one. So the 532 green is the standard, and we've also got a 785 optional laser as well. So here I'm going to compare FTIR data with O-PTIR data collected in reflection. I'm going to point out that the O-PTIR data were collected in about 5 seconds with a half a micron spot size. So yes, there it is. So FTIR, these reference measurements were collected in transmission off a thin film, probably about 10 microns. And the O-PTIR measurements I'm going to show you in a second were collected off a thick block, some millimeters, in reflection mode. Right, so here we go. That's red is the FTIR reference, black is the O-PTIR reference, and you can see it's a spot-on match. Same thing for polyethylene, PET, nylon, and polystyrene. Now, I find this is a really important slide because it really goes to show how the two techniques are comparable and compatible. So data collected with O-PTIR in reflection mode is indeed fully comparable and compatible with FTIR data collected in transmission mode. Spatial resolution is something that we often talk a lot about. Of course, it's perhaps our biggest point of difference, but it's important to understand the basic concepts behind spatial resolution. And that is, what would be an approximation for that is this so-called Rayleigh criterion, which really simply says that spatial resolution is proportional to wavelength in microns times some factor divided by the numerical aperture of your objective, which is the property of the objective. So if you considered a typical FTIR microscope, if you plugged in some numbers at 10 microns, which is 1,000 wave numbers, your spatial resolution is going to be about just over 12 microns. But because this number is variable, if you were to plot that, of course, out here in the short wavelength end, the high wave number end, spatial resolution gets a little better. But out in the fingerprint region, where the most interesting stuff lies, your resolution is typically 10 plus and could be pushing up that 15 microns. But with O-PTIR, it's actually very similar to Raman in the sense, because that probe wavelength is constant, and it's a very low number, it's a good thing, your spatial resolution is low, which is a good number, 416 nanometers, and it's flat. It doesn't matter whether you're measuring at 1,000 wave numbers or measuring out here at 3,300 wave numbers, the spatial resolution is constant. And so that's where that approximate 30x improvement over existing FTIR QCL microscope comes from. Okay, but one of the most exciting things, I think arguably the most exciting thing, is the fact that we can now do simultaneous infrared and Raman, and it's an absolute world first. So same spot, same submicron resolution, done at the same time. And how does it work? Let me take you through this schematic here. So if we start things here, that's our QCL image of that. So we shine that through our objective, and that's your reflective Cassegrain style objective, one that you would find on an FTIR microscope. At the same time, we shine in our probe, let's say it's our green one, this one, and we focus that also onto the sample. This is where that photothermal magic happens. The reflected green light would come back up and be detected with a visible detector. So this is another important point that I sometimes forget to mention. Even though we excite in the infrared, once we've excited the sample, everything downstream from that happens in the visible. So all of the infrared information is collected with a room temperature, high sensitivity silicon detector, photodiode. Now, so that's all the reflected green light. But remember, we're using a Raman grade green laser, which means there's Raman scattering happening all the time. So those Raman scattered photons, which are slightly shifted in wavelength, are separated out by a dichroic filter. So only the Raman shifted photons will be sent off to a Raman spectrometer. And you can see a slightly different color to the green there. And the unshifted, so the Rayleigh photons, will be sent to the visible detector for infrared signal extraction. So in this way, it's actually a very clever, very efficient means of collecting simultaneously infrared and Raman from the same light source, from the same visible light source. So this really takes full advantage of the complementarity we've been taught for decades now. It's confirmatory, so the IR can confirm the Raman result, and the Raman can confirm the IR result. And it's IR, and it's really two techniques in one. So it's a far more thorough, gives you a far more thorough sample characterization. Here's where I'm going to take you through some application overviews. But first, I want to quickly highlight something we're very proud of, the fact that our publications are on the rise. And by the end of the year, we should be getting close to about, I suspect, 30 publications. And you can view all of these on our website. But one that's just come out in early November of this year, so really literally a few weeks ago, and this is a huge one, this impact factor of 39 in Nature Nanotechnology, was all about how baby bottle teats or nipples can release hundreds of thousands, in fact millions of silicone and elastomer microplastics just through the act of steam sterilization. This is something that really caught these researchers by surprise, because these materials are chosen because they're thought to be resilient to this cycle of steam sterilization. Turns out they're not. So the silicone nipples and teats become etched. They flake off and they shed elastomer microplastics. And in fact, so these are some really pretty good quality data that are collected in reflection mode off these sometimes rough surfaces. In fact, even on in one of their figures in the supplementary section, they showed how they were collecting O-PTIR spectra from 300 nanometer polystyrene spheres. And you can see going from 1 through to 7, those points are 1 through to 7 there. So you can see there's 3 spectra that have a hit for polystyrene, and then a couple over here that are on polystyrene, and with nothing in the middle, number 4. Right, so in the conclusion, they stated that they, using O-PTIR, they were able to demonstrate microplastics down to 600 nanometers. And so now, with this sort of tool, it opens up the study of micro and nano plastics. So MNPs is a new acronym. This was another pretty hot publication that came out maybe now a couple of years ago, looking at imaging of polymorphic amyloid aggregates directly in the neurons themselves. And that was a huge one. That was impact factor of 16. And I like this figure because it actually shows how from point 1 to point 2, they're only separated by 282 nanometers. You can actually see protein secondary structure differences. Just to give you a bit of a breadth, so now polymers is another hot area for application for this sort of technique, and IR+Raman is a natural fit there. And in this particular example, we looked at a PHA PLA. These are biopolymers, biodegradable polymers, and we looked at these laminates, a PHA on PLA. These should not be particularly compatible, but they were, so that was the point of the investigation, and particular focus was this interface. So taking a line profile across here at 100 nanometer steps, we collected O-PTIR and Raman spectra, and in the paper they've gone on and have done some 2D correlation analysis. But just a simple demonstration, these are the IR data, the O-PTIR data. So each spectrum is separated by 100 nanometers. And you can see different peaks from different areas using the properties of the QCL, the 725 and the 1760 peaks were imaged. Did some false color maps on that. I thought it was interesting. This here is a car paint cross-section. The reason why I'm showing this is a nice demonstration of where, when Raman fails, because of fluorescence, O-PTIR does not. So if you take a look at these points, red, green, and blue, in 3 different layers. We've got O-PTIR spectra here now, so collected with a QCL and the high wave number OPO, and simultaneously they were collected with Raman. So you can see, though, that the red and green points suffer greatly from fluorescence. So fluorescence will impact our fluorescence measurement, our Raman measurements, or any other Raman measurement for that matter. But O-PTIR is completely unaffected by fluorescence. And you can see that the red and the green with these 2 O-PTIR measurements were completely unaffected. So if you only had Raman to deal with here, you'd have no chemical information. Whereas with O-PTIR, you're never hindered by fluorescence. Here's a defect measurements on, these are hard disk surfaces, in fact, it's courtesy of Seagate. These were pretty thin and small. I think these were like 60 nanometers in thickness, and some also maybe 100 or so nanometers or a few hundred nanometers in lateral dimension. And these were easily identified, so they're pretty happy. Fibers we can measure, so I'm going to go through these pretty quickly, whether it's a 20 micron fiber or a nano fiber. These spectra are all completely unprocessed. So if you've ever tried to measure a spectrum from a fiber before, you'll know that in a traditional FTIR microscope and reflection, it just doesn't work. Another polymer example, again, I'll go through pretty quickly. Quick coarse IR measurement versus an ultra high resolution 100 nanometer step size measurement shows lots of little domains and big domains, and clearly resolve spectra as well from that. And the following slides, you can see some of these are as little as 275 nanometers. One of my first measurements, in fact, I did when I joined the company, is one of my own cheek cells in water, just sandwiched between two calcium fluoride cover slips. And then, in fact, because I was relatively new, I didn't actually collect a visible image, kicking myself for that now. But just clicking around, imagine this was a visible image. I clicked around and took some spectra. I saw some had lipids, some had clear nucleic acid features, and of course protein was everywhere. So I took those three wave numbers, did 3 single frequency images, combined them as a RGB overlay, and wow, I thought that looked just like a fluorescence image. So you can see obviously the nucleus in the middle, and you've got lots of lipid inclusions around the sides and the cytoplasm. And some of these lipid inclusions are sort of half a micron to a micron in diameter. And all these spectra here are inclusive of water. So this is actually a water measurement, and we haven't done any water subtracting. So you can see there is a water peak superimposed on the amide one, of course. These were some breast tissue calcification, but some of these were sort of half a micron, so about 5 microns in size on average. Hence, they were actually missed by traditional FTIR. Flicking back to microplastics for a second, of course, a very hot topic. This was pretty impressive test measurements or proof measurements. We took a few 500 nanometer beads, and you can see them on calcium fluoride here. And spectra collected in seconds simultaneously. You can see O-PTIR measurements in red, green measurements are the Raman measurements. But the important point from this that I wanted to really stress is that whether you're measuring from a half a micron particle or a cluster of polystyrene beads measuring about 4 microns, the spectra don't change. So O-PTIR spectra are robust, they're repeatable, and they're independent of particle shape and size or otherwise sample morphology. Okay? And here's in fact a real-world microplastic sample on a gold-coated polycarbonate filter, so you can actually measure microplastics directly on the filters through which they're being filtered through, and that's a 7 micron particle easily measured of polycarbonate sorts of things. And because now you're collecting infrared and Raman simultaneously, you can now search these databases simultaneously as well. And with Know-It-All, now handled through Wiley, you can actually plot. This is a 2-dimensional scatter plot, the heat quality index. So the upper right-hand quadrant is one where you have a high infrared heat quality index and also a high Raman heat quality index. So that also that comes back to the confirmatory aspect rather of IR and Raman measurements. This is a paper by Andy Alt looking at atmospheric particles, again infrared and O-PTIR, including even the OPO measurements here. Kathy Goff from University of Manitoba in Canada looked at fibrils and tissues, compared that with FTIR and even with nano FTIR to show that they were very equivalent. And then, in fact, O-PTIR could do polarization measurements both in the parallel and in perpendicular that the nano IR could not. In fact, she went on further to even compare glass, calcium fluoride, and glass measurements. And this is one of the exciting aspects that I'll touch on a little bit later as well. That materials, that tissues tissues of perhaps several to 10 microns plus put on glass work completely, and there's absolutely no glass contribution to them. This in fact, I should update this because the paper has in fact been published, was some work I did with Keele University, looking at cell differentiation, cancerous and well, malignant cells and normal cells using glass substrates. So these are some spectra. These are single scans. That's about 1 second measurements from a 0.5 micron spot on cells on glass. And with very thin materials, you do see a bit of a glass contribution. But from about this point onwards, you're completely unhindered by glass. And even over here, you could possibly subtract out the simple broad silicate peak there. But this is really the first example of unblocked, completely unblocked glass substrate measurements in the infrared. Submicron features in these images were easily detected. And this was collected using one of the newer CH fingerprint QCLs. So over here we had a CH, rather a lipid chain length measurement. We looked at CH2 ratio to CH3. Of course, looked at the protein. It's a pretty simple measurement as well. These are 100 nanometer step images. Peter Gardner at University of Manchester published a paper also recently looking at live and fixed cells, the live cells being in water. Normally, of course, this is pretty difficult with FTIR, if not impossible. You have to work with very difficult and narrow path lengths. But they demonstrated that O-PTIR in this upside-down sandwich configuration, where the cell is actually upside down and where the infrared hits the cell first, hence it's not attenuated by the water, this actually works really well. And they were able to show, clear organic, organelle features of live cells, hydrated cells, and they compared that with also some fixed cells too, using both infrared and Raman, and also infrared with the OPO laser. I think this is my last paper example. This is from the Roy Goodacres group at Liverpool, and they used O-PTIR now to target single bacterial cells. And this is something that's never been done before, because regular IR could never resolve single bacteria. And they did, and they fed these bacteria different isotopes, heavy carbon, heavy nitrogen, and that has a huge impact on the amide 1 and also the amide 2 signatures. And from that, you can learn a lot about the metabolism of the cells. So, like some of the samples I borrowed from them, I've gone back and done some measurements myself. So this is in fact a measurement with the silent region QCell. You can actually see these see some CD bands. These were E. coli grown in deuterium or heavy water. And this is an image of a single E. coli bacteria collected at 50 nanometer step sizes. This is infrared far-field imaging we're talking about. And my last example is again of E. coli. I think this one is not deuterium labeled, but what I like about this is that this was one of my early simultaneous IR and Raman measurements. So here we're getting infrared and Raman from a single cell at the same time, and this was about a 20-second total integration of both the infrared and the Raman. It also shows just the inherent sensitivity advantage of that IR has. You can see that the O-PTIR signal is orders of magnitude higher in sensitivity in signal noise than the Raman measurement. All right, so really my clear takeaways is that with O-PTIR we're pushing infrared microscopy beyond these traditional and what are I think even accepted limitations. We're now combining IR and Raman into a single platform. Obviously with the submicron resolution you'll see a lot more. It's non-contact. You don't have any of these dispersive scatter artifacts which have been such an issue with biology, with biospectroscopy, and having to correct for all that me-scattering. Little to no summer preparation, lots of new QCL options opening up new spectral ranges, and recently we're also starting adding fluorescence modules as well, so you can actually now do co-located fluorescence O-PTIR. And I'll finish with this again, same spot, same time, same resolution with infrared and Raman. Okay, everybody, so we've now come to the end of my talk, and we've also come now to the main speaker of today's webinar, and of course that's Dr. Christoph Krafft from my PhD in Jena. And I'll let him introduce himself and let him introduce his own presentation. Christoph, over to you, please. Thank you.
Yeah, hello, everybody. It's a pleasure for me to talk in this webinar from Photothermal about our recent experiments on live cells, tissue, and single cell bacteria with submicron and simultaneous Raman using the Mirage. So in the talk, I will focus on the benefits and for biomedical studies.
Right.
Yeah, so I will start with my, in the first part with my personal experience in infrared spectroscopy, and then describe some key experiments for bacteria, cells, and tissue. I compare the O-PTIR results with FTIR spectroscopy, but here in the context of tissue, also with Raman in multiphoto microscopy. And in the end, I will give a summary. So I work with infrared Raman spectroscopy since 1994, when I started my PhD in Berlin. In 2001, when I started my position in Dresden, I was one of the first users of a Bruker Hyperion FTIR microscope with a 64 by 64 focal plane array detector. And I will refer also during my presentation to these previous results of the studies. For example, here the identification of B and T cells by FTIR microscopy. 2009, I moved to Jena, and I was able to purchase a FTIR imaging system from Varian, which is now Agilent, and this is also this was also equipped with a 64 by 64 focal plane array detector. Here are some photos of the instrument in the lab. Here, that's the Agilent system, and this was the former Bruker system on the right. And last year, finally, we were able to order the Mirage instrument from O-PTIR, and it was the first instrument with 785 probe laser. So, and all data that I present today were recorded with this O-PTIR instrument. Yeah, the first topic, the microbiome identification by infrared spectroscopy, state of the art is that an FTIR spectrometer is connected to a high throughput accessory. And then standardized 96 and 384 well microplates can be used, and for infrared spectroscopy, these microplates are made of silicon, zinc selenide, and germanium, and some of them are shown here at the bottom. The workflow is that a small volume of liquid sample between 1 and 20 microliter is pipetted on the wells, and then they are dried, and then a single spectrum is measured per well. There are infrared spectral libraries available with more than 100,000 compounds, and there are also libraries available for microorganisms. The O-PTIR perspectives. First of all, quantum cascade laser are used as radiation source, and this enables new detection schemes, the so-called optical photothermal detection, as explained by Mustafa, with submicron resolution, and this enables to collect spectra on a single cell level, that means from single bacterial cells. Yeah, there are also less expensive options for microplates, for example, glass microplates, which are coated with glass, with metals, and the workflow can also be changed. So liquid samples can be probed, and so no drying is required anymore. Yeah, and then instead of a single spectrum per well, a full image can be measured per well, and existing FTIR spectral libraries can be applied for chemical compounds, and for microorganisms, the transferability of a library needs to be shown. But preliminary data are very promising, as shown on the next slide. So here we compare FTIR and O-PTIR spectra of bacteria, and it's straightforward in an FTIR image. Here to extract a bulk spectrum, so a certain area can be selected, and then an average spectrum can be calculated. And here 3 average spectra are shown for our use to compare the Bacillus subtilis, that's red trace. Bacillus pumilus is a blue trace. And Escherichia coli is a green trace. And the low with a shoulder. And the two band is quite similar. A single bacteria cannot be resolved in with the standard configuration of this FTIR microscope. The bulk spectra of the O-PTIR instrument share many similarities with the FTIR spectra. For example, here is the shoulder of the M1 band, and also here the three features in the low wave number range are very similar to the FTIR spectra. But the main advantage O-PTIR image was collected from a couple of single bacteria, and from several positions also full spectra were collected. And if you take a close look, you can, for example, here the three spectra from this bacterium show some variations. For example, here the shoulder in the one band has a different intensity, and here for three other bacteria the shoulder is more intense. So there are variations, and that are evident in these first data, and these need to be studied in more detail in the future. So the next topic is FTIR spectroscopy and imaging of single cells. The state of the art for a couple of years ago was to use synchrotron radiation with a small aperture, for example, 8 by 8 micrometer. If the aperture is in this size range, the throughput is very small, and overcome the problem of the low intensity, synchrotron radiation can be used. Single channel is used, and then quite a high number of scans per spectrum, and then here a single cell can be scanned. So an FTIR imaging microscope was a progress. In 2007, I published a paper about mesenchymal stem cells. So such a system used a glow bar as an excitation source for the infrared microscope. Aperture is not needed because the segmentation is achieved by these 64 by 64 pixel focal plane array detector. Each pixel corresponds to an area of approximately 4 by 4 micrometer. In these experiments, 20 scans per image were collected, and a mosaic of 4 by 4 images. And in this field of view, approximately 100 cells were present. Yeah, in the confocal infrared microscope, the lateral resolution is approximately 20% better. That means resolution limit around 7 micrometer. However, the sequential registration gives a quite low number of spectra, and also a smaller region of interest compared to this FTIR microscope with a focal plane array detector. In this paper from 2007, I studied mesenchymal stem cells, and I found 4 classes of cells before osteogenic stimulation. That's the spectrum in panel A. After, and then there is a subclass of in this image where the cells accumulate glycogen. Then after osteogenic stimulation, the infrared spectra changed. Here, the most prominent change is small lower shoulder of this M1 band. And then after stimulation, also some cells express matrix. That means hydroxyapatite. And this is also clearly visible in the red spectrum. So, and LDA discrimination model was trained by 2 training data sets. So red is unstimulated class, yellow the glycogen class, blue the stimulated class, and cyan here stimulated class with hydroxyapatite, and another data set was collected as for training. So what can O-PTIR do better? With O-PTIR, we studied also a mesenchymal stem cell, however, with a much in a smaller region of interest and with a step size of 100 nanometer. And then certain subcellular features are resolved. First blue, that's the cell body. Then the cell nucleus is resolved, and even the nucleoli in the center of the cells is also evident. And the dimension are shown here. So the cell body is something like 24 micron. The nucleus something like 6 micron. The nucleoli 1.4 micron. And then the delineation zone, the membrane-like structure, is just something like 200 nanometer. These are representative single-cell spectra. That means green, the prominent glycogen feature, makes up a large portion of the cell. And also here, the membrane-like structure gives a clearly different spectrum than the cell body of the surrounding cell. The conclusion is that the better spatial resolution in the submicron range not only generates high quality, more information-rich infrared images, but also delivers purer infrared spectra from the measured locations. So in another series of experiments, we measured O-PTIR microscopic images from cells in aqueous buffer. So the first cell line was a leukemia cell, THP1. So cells were grown with 80 to 90% confluency. We pipetted a small amount, some around 4 microliter, between two very thin calcium fluoride slides. The thickness was just 200 micrometer. Data were collected in transmission mode. And of course, the data that are shown here are shown without water subtraction. That means this amide 1 band contains also spectral contributions from water. And then, so this is a mean spectrum and standard deviation. So some spectra have a high content of this ester band due to lipids. And then a representative protein band is the amide 2 band. And then also further bands can be assigned. The images, the blue channel is a protein distribution, and the red channel is a lipid distribution, and even very small lipid feature can be identified. For example, here this small lipid droplet only has a diameter of 670 diameter. Yeah, we also collected more data from other cells. For example, in HeLa cell, that's the cells derived from the cervical carcinoma. This HePa-RG cell line, that's the liver cell line, Raji lymphoma cell lines, and then 3 different leukemia cell lines. Altogether, almost 8,000 spectra are collected, 5 spectra per cell, and then we used an average spectra for classification. Data were collected on 10 days, and 70% of the data were used for training, and then 30% of the data were used for testing. And on each day, all cells were studied. Yeah, the first day, the first model distinguish here this non-leukemia cells, and PLS LDA model nicely discriminates the 3 cell lines. The model 2, the discrimination of the leukemia cells, there is more overlap visible. And then the third model was trained, which discriminated leukemia versus non-leukemia cells. So the training data are very well separated, but also the test data show only a small overlap in these whiskers, and the majority of cells are nicely discriminated. So it's possible to use O-PTIR and spectra to discriminate cell lines, and we are working on better cell sample chamber to continue these experiments. Next topic is FTIR-Raman and multiphoton imaging of brain of tissue section, and I as an example I show the application to identify the primary tumor of brain metastasis. In a previous publication, we developed a hierarchical support vector machine classification to determine the primary tumor. In the first step, we identified the carcinoma spectrum in red from a Raman image and from an FTIR image, and these data were subjected to a second support vector machine to identify the primary tumor, and the blue color indicates that the primary tumor is a lung carcinoma. And both Raman and also infrared spectroscopic imaging gave a similar classification. It seems that Raman spectroscopy has a better lateral resolution because these tumor cells here in this necrotic portion of the tissue can be better resolved than in infrared imaging. The CAS image was also collected from the same tissue section, and also here the margin is clearly identified, and the CAS microscopic image was tuned was collected at 2850 wave numbers, which is which probes mainly lipids, and the main discrimination here, the main contrast is due to lipids, and CAS offers also cellular contrast. Here these these dark spots corresponds to single cell nuclei, and these two cells are typical for tumor cells, and these tumor cells are also clearly resolved in this CAS image. Back to O-PTIR. Of course, the O-PTIR can also probe lipids, and here lipids are also very prominent in the bladder tissue section. So there are large lipid droplets in these two images, but also very small lipid droplets. So, and the lipid droplets can clearly be identified in the tissue matrix, which is here in the green channel. And also full spectra have been collected at selected position. So the region of interest was here 400 by 400 micrometer in the top image and 1 by 1 millimeter at the bottom image. And the spectra are virtually background free. That means without further correction, the baseline is close to 0. A problem for FTIR microscopy is lung tissue, but because it's a very porous tissue, as shown here, there are holes which are assigned to bronchioli and alveoli, and here infrared image was collected in the macro chamber. So this corresponds to 4 by 4 millimeter. Each pixel represents 62 by 62 square micrometer. And here from this area and this area where this tissue is quite homogeneous, reasonable spectra with a low background were collected. However, here at positions 3 and 4, where the close to the margin of the tissue, the spectra are strongly affected by anomalous scattering. The baseline might correct the background, but it's more difficult to correct the shift of the band. So instead of the expected position of the M1 band at 1657, the M1 band at these positions are shifted to 1649 or 1646. We studied also lung tissue from mouse by O-PTIR, and it's clear that the O-PTIR spectra do not suffer from scattering artifacts. This is evident here by two representative spectra. We collected also a multi-photon image from a region of interest. So the CAS channel is here plotted in red. Two-photon excited fluorescence is plotted in green, and secondary harmonic generation is plotted in blue. And for example, here around this bronchiole, there's a ring of a strong second harmonic generation contrast. And such ring is also evident in the O-PTIR image when the ratio of the amide2 versus amide1 band is plotted. Interesting is also the ratio of these lipid band 29-22 versus the amide1 band. Then here a nice contrast is obtained for this feature, which is also, it has a high two-photon excited fluorescence contrast and also a high CAS contrast. So there is a nice agreement between these features in this multi-photon image and the O-PTIR image. In the O-PTIR here, the interior of this bronchiole is filled with material, with dried material, and this is quite weak in this O-PTIR image. And probably the sensitivity is higher of the O-PTIR imaging approach. Yeah, the last example is infrared imaging spectroscopy of spleen tissue sections. So this was one of my first study using FTIR imaging, and so of course we studied unstained tissue section, and of course, and the contrast is very low. So a parallel tissue section was prepared, and the B and T cells were stained, and this gives a contrast. So the FTIR image was segmented by cluster analysis, and here the gray cluster for the follicles was further analyzed by a second cluster analysis, and this contrast nicely agrees with the immunohistochemical staining. From two follicles, a microscopic image was collected, and the spectrum, infrared spectrum corresponding the orange cluster, was assigned to the red pulper. It's a prominent feature in spleen tissue. Numerous cracks are evident throughout the tissue. These cracks, if you look closely, also show some anomalous scattering and a shift of the amide1 band. And so the red pulper and the cracks were removed in the second cluster analysis. And the second cluster analysis shows here a ring of white pulper with B lymphocytes, and this spectrum shows very intense nucleic acid bands. So we also, from our clinical partner, we received some spleen tissue section from a mouse model, and these sections were first studied by multi-photon imaging. Again, CAS is a red channel, two-photon excited fluorescence, with a green channel, and second harmonic generation. So these samples were prepared on a quartz slide for infrared spectroscopy. The samples were prepared on calcium fluoride slide, and but we were able to identify the same follicle. There's a small change here in this area, but here the follicle is clearly visible, and the follicle is also visible in the O-PTIR imaging by plotting the ratio of this 1086 band and the M1 band. This image was collected pixel size of 1 micron per, and the time was approximately 7 minutes per wave number. That means for these two wave numbers, this was approximately 15 minutes. And the image can also be overlaid with a visible image to highlight the area with the white pulper and the follicle. From a small region of interest, ultra-high resolution infrared image was collected. And here it was very interesting. So here the pixel size was again 100 nanometer per pixel. And the spectra were normalized. No other post-processing was required. It took approximately 5 seconds to collect a single spectrum, and the spot size was 500 micrometer. So spectra were collected from the spot with a high intensity of these 1086 band, and also the band at 1244. It has a very high intensity, and then here the red spectra were collected from the blue colored areas, and these spectra have much lower intensities at 1086 and 1244 relative to the M1 band. And from the size of these features with a high nucleic acid content, it's approximately 4 micron in diameter, and the infrared spectra with these intense nucleic acid bands, we conclude that single cell nuclei were resolved in these follicle of white pulper in spleen. And the identification of cell nuclei and tissue section was in a former study not successful by FTIR imaging. So in this study, we notice here some in the unstained tissue section of liver, we notice a few spots, and we couldn't assign the spots, so we collected an F2R image and also Raman image. And in Raman image, we could resolve features which high intensity of nucleic acid bands, and these features correlated well with the cell nuclei. However, if you take a look in the infrared images, FTIR images, single cells or single cell nuclei couldn't be resolved. Also here the spectra are affected by scattering around the cracks, and also here there is a high scattering at the margin of the tissue, and we used for follow-up studies of brain tumor section Raman spectroscopy. So the cell nuclei is a very important feature for pathological inspection of tissue section, and it can only be resolved by O-PTIR imaging and of course Raman imaging. So I come to the summary. The benefits of optical photothermal infrared spectroscopy for studies of bacteria, cells, and tissues. First of all, the data are thickness independent if the spectra and images are collected in reflection mode. If the samples are thin enough, so between 10 and 20 micrometers, they can also be studied in transmission mode. Images can be rapidly collected at selected wave numbers. Rapidly means depending on the size, in a minute or a few minutes. And full infrared and Raman spectra can then be collected at selected position. The images and the spectra offer submicron resolution, no dispersive line shapes, and no rescattering effects are evident. There's a wide choice of substrates compatible with O-PTIR. First of all, calcium fluoride, mirror IR. These are reflective slides. You can use metal substrates or even inexpensive glass substrates. If the samples are thick enough, that's no problem. The data are less water sensitive due to the optical detection. So and this offers great promises for cell studies, and also polarized infrared data, and are possible because the QCL laser is polarized. So I'm very happy to use the instrument, and I will publish more details in the next months about the examples that I presented today. Thank you for your attention.
Oh, great. Thank you, Christoph, for that really interesting journey you've been on. Looks like there's a lot of great work coming our way. Look forward to seeing those published. Now, I've been sitting here watching lots of questions come in. I think the first one, though, I'll take for myself. The first question says, if O-PTIR uses a green laser for both Raman and O-PTIR detection, why does O-PTIR not have fluorescence when Raman still does? The key to that is the fact that the IR signal is modulated, it's pulsed. And so that means that the infrared photothermal signal is also pulsed and modulated. And that appears in the green reflected light or transmitted light signal as a slight modulation in the amount of visible light hitting the detector. And we use the lock-in amplifier to remove and extract that modulated signal, which contains the infrared information, whereas fluorescence is not modulated. Fluorescence only comes from the visible laser. So in that way, we very simply and easily remove the fluorescence, which unfortunately still impacts the Raman signal, but has zero impact now. Even if it's at the worst fluorescing sample in the world, it will have absolutely zero impact when it comes to the infrared signal. Christoph, the next one I'll give to you is, do you see the possibility to do rapid bacterial ID at the single cell level? And will the natural cell-to-cell variability be an issue? What do you think?
Yeah, the classification model need to consider the cell-to-cell variations. And in our institute, we have a group that developed classification models and to identify bacteria using Raman spectroscopy. And so, but we trained the models ourselves by cultivating the bacteria in different conditions, and also in a similar way, also the cell-to-cell variability need to be considered in these classification models. Therefore, I put a question mark if the transferability of the FTIR libraries for bacteria is possible. So the effect of the cell-to-cell variation need to be studied in more detail. Yes.
All right, thank you. But I think for me, I'm excited at the prospect of now not having to culture bacteria. So I think that the potential now exists to be able to filter directly bacteria out of various bodily fluids or other fluids, and to be able to measure the cells directly. I think it sounds like it'll be a great saving in time. Let's move on to the next one. This one I'll give to you as well, Christoph. How do you see the value of IR and Raman working together for these sorts of applications?
Yeah, of course. Both infrared and Raman spectroscopy, they are complementary to each other. For example, there are in Raman spectra a lot of signals from aromatic amino acids, which are almost invisible in the infrared spectra, and therefore these two spectra are highly complementary. Also, so-called two-dimensional models can be trained and developed using one dimension for the infrared spectra and in second dimension for the Raman spectra. And I think the combination of these two modalities, infrared and Raman, would give much more accurate classification models. We will also try to explore these possibilities in the near future.
Thank you. In fact, the next one I might take for myself. It says, you mentioned new substrate options opening up and being able to measure glass substrates. And it says, how do you see this working? So I've actually done some a recent publication using glass as a substrate for single cell classification of cancers and non-cancerous cells. And it's really interesting because now, for the first time really ever, you can do infrared spectroscopy on glass. There are a couple of caveats though. If you want completely unhindered fingerprint spectra in the infrared without any glass silicate peaks coming through, you probably need something that's a typical histological thick section, like a 7-10 micron plus type of section. And when you have that, the infrared doesn't penetrate enough to the glass to generate a photothermal response. So for tissues, I think it'll work really well. In fact, I know Kathy Goff has published a little bit on that as well from Canada. But the work that I did looked at thin single cells, which are probably 1 or 2 microns. And so when you're working at that thickness or thinness, glass does have a silicate peak which comes through. And though I didn't ever explore subtracting that, so I think the potential exists to be able to subtract that out and still make use of the features underneath the nucleic acids and the carbohydrates. But even if you don't subtract it, and that's what we did with the paper, we didn't subtract, there's still a lot of information in the protein region, which was more than enough, and also, of course, out into the CH region as well. So that was actually more than enough to achieve excellent sort of high 90s classification accuracies. I'll move on quickly. This is probably for you. It says, were the spectra you showed of the stem cells on O-PTIR class averages or individual spectra?
The O-PTIR spectra were individual spectra from a single point. So that means a single spectrum from the glycogen, a single spectrum from the cell body, and also a single spectrum from the 200 nanometer membrane.
Okay, I think we're getting to the end here. What is meant, in fact, let me take this one. What is meant by purer spectra, and how is this related to spatial resolution? All right, so this is something that I get quite kind of excited about, because we talk a lot about spatial resolution. That's quite clear in the images, in the fact that you can see smaller things. But there's an added benefit which I think isn't always obvious, and that's when you have that higher resolution, it means that you are now collecting spectral information from a very small spot. So that small spot means that you're not averaging over a large spot that you would be for, say, a traditional FTIR or QSL type of measurement. So your spectra are in fact more pure, especially if you're measuring an inclusion that's, say, half a micron in size, 500 nanometers. You can now get spectra, chemical information, only from that one spot, hence pure spectra. Whereas if you did that with a traditional IR system, you'd be averaging over several microns. And hence, I call that sort of spatial blur coming into the spectra. Maybe we'll make this the last one. This one's for you, Christoph, I think. What do you see as the advantages of measuring cells in water rather than fixed and dehydrated? And that they say that it would be interesting to compare classification of cells in water versus dry. What do you think?
Yeah, when you dry the cells, many biomolecules denature. So the proteins and also nucleic acids denature. And for nucleic acids, it is particular bad because the nucleic acid bands give much weaker in the dehydrated state, because change the conformation from the normal B form to an unordered form. And this gives a much weaker intensities of the nucleic acid bands. And so it's more difficult to resolve the cell nucleus in dried cells.
Okay. I did say it was going to be the last one. We've had one more come in. Now this will be the last one. In fact, I think I'll take this one. How is it that O-PTIR does not suffer from knee scattering, but FTIR does, and does Raman too? I think the simplest way I can try and explain this, and of course knee scattering is a very complex phenomenon, but one that's very obvious in the spectrum, and it comes from the fact that in regular IR, so that's FTIR and some of these other Q-cell techniques, you've got these wide range of wavelengths, all of which are interacting with the edges and the particles in very different ways and scattering in different ways. So it's the plethora, it's the broadness of the infrared radiation, in which is a negative in this case, because you're also detecting on the infrared. And that's what creates this wavelength-dependent scattering, and that comes up, and that really upsets the spectra. Whereas with O-PTIR, using a fixed detection or probe beam, 532 typically, or 785 in Christoph's case, so it's single wavelength, so that removes that wavelength-dependent scattering artifact. And Raman is the same as well. Of course, Raman doesn't also suffer from these scattering effects. And with that, Christoph, I'll thank you for taking the time out of your busy day. I much appreciate you sharing your reflections and your sort of future views of this technique. I want to thank the audience for their time to dial in and listen to us. This will now be on demand. It's now recorded and will be available for others to see. So we'll be sending out emails to those if you want to share the link. And if there's any questions that have been left unanswered, we'll do that via email in the next few days. And with that, let's bring this to a close. Once again, thank you to everybody. Have a good day, everyone. Bye-bye.